and which causes mortality. So as the result, the medicine will become ineffective. So the infection will be always persisting in our body and giving the risk to spreading to the others. This is the definition what WHO has given the term for AMR. So what will be the newer threats? We all know that AMR has emerged as a new global threat. So when we are going to use the inappropriate use of antibiotics, which has resulted in more and more antibacterial strains or more bacterial strains developing resistance over the last few decades, and that will cause the fatal consequences. So AMR has adversely affect the prevention and treatment of the infectious diseases. So it may complicate the therapeutic management of diseases. You know that the treatment which we are going to give for infectious disease will be the example are HIV, malaria, in compromising chemotherapy also. So it will make the compromise in this aspect. So we all know that the discovery of antibiotics has laid a strong background and strong foundation for the treatment of effective treatment of infectious diseases. But in reality, the control of these diseases like cholera, typhoid was not possibly due to this of antibiotics. So the technique like pasteurization and disinfection has contributed more and more. So vaccination and antibiotics will not be responsible for eradicating these type of diseases such as smallpox and all these things. And another more thing, we all know that multi-drug resistance in TB has been seeing more and more day-to-day life. So what WHO has stated is around 6 lakhs people have developed multi-drug resistant TB over globally in 2018. It may increase in future also. So when we are going to implement poor regulation in these type of drugs, when we are going to prescribe some of the drugs which we are not having the adequate knowledge among the medical practitioners or some other as well as activity or anti-inflammatory or modify the immune system according to the pathogens. We all know that plant metabolites or the secondary metabolites like alkaloids, polypeptides, flavonoids, or flavonoids for this type of antimicrobial activity. So, which are having the antimicrobial activity, we can target through its synergistic effect because we all know that bacteria formulation or metallic formulation strongly to target these type of pathogens. So, what are one will be having the doubts whether these type of things have already given in Siddha text or not. Enteric infection, skin infection, anorectal infection, and then sexual. So how we can prevent these type of Siddha system? That also mentioned in Therayar Pini Anuga Vidhi, which we have to do and which we have to do not follow this type of lifestyle so that we can have disease free life, a disease free life. So what are healthy living way? We everyone should have proper and one more thing is in Siddha system there is a practice of applying herbal hit. There is a practice of applying herbal hit. Use of applying herbal help prevent the infectious diseases. So again, there are in Siddha. What we all know that rejuvenation and longevity and all their antimicrobial which will reduce the incidence to develop these. Sorry.
Hello.
So, sir, good morning.
Ma'am, very good morning, ma'am. Thank you for your wonderful presentation
Thank you, sir.
for enlightening our knowledge. Ma'am, I have some doubt like what is the now rules and regulation for the herbal medicine should be used for this drug resistant pathogen? Any rules and
Over the counter, sir, because some of the drugs are there. If I am going to give Lingachanduram, which is having the strong antibacterial activity against E. coli, Salmonella aereus, or Klebsiella, or Salmonella typhi, or Vibrio cholerae. These are all the things which we can give Lingachanduram, because Lingachanduram is having the mercurial drug, so it cannot be given as such to the patient. Some of the things are there. We have to be very cautious when we are going to use these type of scheduled E drugs. We cannot go and take nowadays. It is everyone is suppose if anybody is having upper respiratory tract infection or lower respiratory tract infection or any urinary tract infection, everyone is going to the pharmacy and everyone is asking augmentin or then azithromycin or then ofloxacin or something like that. They can get these type of antibiotics via over the counter itself. But bacterial act for some other viral or something like that. Then so we have to be something like that. They very keen that we have to go and get prescription from the Siddha practitioner.
okay, ma'am. Thank you, ma'am. Ma'am, on the one more thing, if you had without metal, if any, for example, lot of students and other research paper, we can see some herbal plants they are finding it have the capable for the activity, either single plant or
Yes, sir.
multiple plants without metals. If in this concern, if it want to convert as a drug format, what
Yes,
is
sir.
the procedure, ma'am? How CCRS is supporting as a researcher to make commercialize this product?
Commercialization. Yes, sir. For example, I will tell one example. For example, Osimum sanctum. For example, Osimum sanctum is the thing which everyone is known. In Google itself, it is telling that it will cure upper respiratory tract infection. Suppose if we are going to along with Vitex negundo, Nucci, or then Piper longum tipuli, or along with Nigella sativa. These are all some of the antibacterial. These are all having the antibacterial activity. So we can make into a powder form, or we can go for the capsule form. And we will what we can support is we can go for the standardization procedure as per the PLM properties. What PLIM has given is according to the PLIM guidelines, we will make the study and we will give to the students so that students can make the thing, they can go for the license. For that, they have to approach for state licensing authority for getting the license. After that, they can go for commercialization. Or again, if they are not willing to go for the commercialization or something like that, if they are going to commercialize that product, we will give the soft loan of 2.5 lakhs so that they can go for all the type of studies and they can launch the product in the market. After getting profit from the market, they can settle the loan to the CCRS.
Okay, ma'am. Thank you very much, ma'am, for your clear clarification about this. Thank
Yes,
you very
thank you,
much,
sir.
ma'am.
Thank you, ma'am. I was a bit late.
Thank you, sir.
Sai here, ma'am. Thank you,
Okay,
ma'am.
sir.
Thank you for your talk. I guess many people would have got the information on how natural sources can be used as against antimicrobes, as antimicrobes. So even your information of on urinary tract infections and the current research topic which Siddha is currently pursuing, Orai Materai, where many of us have been into it. So I guess our students and many other people who are listening would have got little bit of insights on each and every aspects how Siddha has been involved in combating the antimicrobial resistance. So I thank you personally and on behalf of the Department of Biotechnology, Welltech High Tech, for accepting our invitation and being here for past 1 hour and enlightening our students as well as other research scholars, ma'am. I guess there is not much query being posted and only positive notes were being received.
Okay, sir.
So we can close the session here, ma'am. Thank you, ma'am. Thank you for your time.
Okay. Okay, sir. Thank you.
Thank you, ma'am. Students, the next session starts by 11:45. So people can take a break of 10 minutes or 15 minutes. We'll begin the next section at 11:40. So there will be a short break for 10 minutes. Thank you.
the diseases, or what is the deadliest among whatever is present on this earth, and if you go with the statistics, it's the mosquito, basically, which is the deadliest animal, which is basically causing a lot of deaths uh, all over the world. And if you talk about mosquito, it is actually a major vector for many of the diseases, like like chloroquine. Chloroquine actually, there are it is not right now recommended for Plasmodium falciparum, but for Vivex, Plasmodium vivex, it is still given. And but the problem which we are facing is drug resistance. So let's say, what is the big deal? Why we want to know about malaria, or what is the problem which we are facing, that even after knowing malaria parasite, that malaria is being transmitted by a parasite known as Plasmodium, there are the vector, we are still having a lot of cases throughout the world, around the world, like 241 million cases were recorded in 2020, which is again said to be underestimated, because at that time, because of COVID, the reportings was very low, and with a lot number of deaths. It is estimated to cause a death in millions of people, and where 30% of this include the children, mainly in African region. It is endemic in 87 countries, and about 95% of malaria deaths are from 31 countries. Until date, we know only one vaccine, Mosquito, is available, and there also we have a limitation of the efficacy. Parasite has, like, a lot of drugs have been tried against parasite, but parasite with time have adapted resistance against most of these drugs. If we talk in terms of what is the scenario in our continent, Southeast Asia, so we'll see. If you see over here, this green portion, India is contributing to almost 88% of the overall cases in Southeast Asia. With,
Excuse me, ma'am.
yes,
Will be, ma'am. Your slides are not moving, ma'am.
oh, it's
Yes,
not
now it's
moving.
now it is moving. Ma'am, I guess you
Oh,
have not shared the screen as a whole. The entire desktop has to be shared.
okay, okay, okay. So sure, I'll share the desktop then. Just a minute.
Yes,
Thank you for telling me. Just a sec. I'll
yes,
stop the presentation and I'll again do it. Okay.
yes,
Thanks for
yes.
telling.
Thank you.
Okay. So I'll just so this is what I have shown over here, that there are so many cases. So uh, okay, just a sec. Now is it visible?
Ma'am, it is visible now.
And now is it moving?
Yes, yes, it is moving also. Yes, ma'am.
Okay. Thank you.
Thank you. Thank you.
Yeah. So basically, as I was saying, that if we talk in terms of Southeast Asia and this green region which you are seeing over here, this is what is representing the reported from Southeast is from India. And if you talk in terms of deaths, also 4% of the deaths are from India region throughout the world. So basically, if you talk about the malaria region, and if the parasites, like if we talk about malaria, the major parasites which are responsible for causing malaria are Plasmodium falciparum and Vivex. The majority of the cases of malaria are caused by these two parasites. However, a few of the cases have been reported from malaria, P. malaria, and P. ovale, with and recently another primate parasite, Plasmodium knowlesi, has also been reported to cause malaria. So if human malaria, I'm talking about the human malaria. And if you talk about these things, so if you see over here that in India, most of the cases are being through Plasmodium falciparum, and similarly, Vivex also has a majority of the cases, and it is widespread. So if you see over here, these are the two main parasites which are responsible for malaria in India. Coming to Plasmodium vivax, if you will see over here, you can see over here that in India, the cases which are there in 2010 and 2019, almost when in 2010 it was almost equivalent, slight increase of vivex cases have been reported in 2019. So we can see that vivex malaria is actually a big problem in Indian scenario. Okay, and most of the cases in India of malaria are mainly reported from the eastern states like Odisha, Kolkata, Jharkhand, northeastern states like Meghalaya and Tripura. And where I stay, Rajasthan has also been endemic for malaria for around 25 years, where most of the malaria cases were reported from Bikaner, which was declared actually a hyper endemic area from malaria after the 1994 epidemic. So basically, if we talk in terms of malaria and Bikaner, if we see, is the region where most of the cases which we encounter are due to Plasmodium vivax. So what are the symptoms which tells you that you are having malaria? So the major symptom which is being said for malaria is fever with chills. You get, then other than that you have headache, nausea and vomiting. And nowadays, with this COVID 19 coming into picture, which are or any other viral fever which has similar symptoms, it is very much essential that the diagnosis should be proper. And the problem which is happening over in malaria is that we are normally seeing a lot of cases of malaria throughout the world, and most of the cases were uncomplicated, and earlier it was only reported that severe complications like your ARDS, renal failure, etc., are only caused due to Plasmodium falciparum, which, if untreated, may lead to death also. But recently, the severe malaria disease has also been recently, I would say recently, in fact, it has been almost 20 years now, that severe malaria has also been associated with Plasmodium vivax, where the complications similar to Plasmodium falciparum, like the cerebral malaria, the renal acute renal failure, hepatic dysfunction, hemoglobinuria, abnormal bleeding, ARDS, severe anemia, all these symptoms are also being reported from Plasmodium vivax, complicating the situation to good extent or more extent. So basically, it's required that we should now start looking more into this parasite, that is Plasmodium vivax malaria also, because for long it was believed that Plasmodium vivax is a benign parasite. But however, the reports of severe malaria or severe complications from Plasmodium vivax have substantiated the need to look into the parasite. In fact, if I talk about this, the first reports of Plasmodium vivax, where it was confirmed that the severe complications are due to Plasmodium vivax mono infection, came from our labs only, where we have a publication in 2005 in Emerging Infectious Diseases, where we have shown 11 cases from Bikaner, which were having complications like cerebral malaria or acute renal dysfunction or hepatic dysfunction, and these all were having Plasmodium vivax monoinfection. And later on, we have reported many cases from the same region where we have seen severe complications due to Plasmodium vivax. So basically, when we talk about the diagnosis of malaria, till date the microscopy remains the gold standard to scrutinize the suspected malaria cases, because it is actually uh, very sensitive. But however, the people have been able to diagnose it properly, because remember one thing, with the development of drug resistance, there has been a great demand of uh, identifying or diagnosing the parasite well, because the drug may be effective against Plasmodium vivax, but may not further be effective against falciparum, and then you need to very well distinguish between the two, because otherwise giving doses which, like giving the drug for which falciparum is resistant to, that thing may develop further resistance as well. Other than that, the commonly used method for the diagnosis is the rapid diagnostic test, where you have the antigens like HRP or LDH, which are being used to identify whether it's a Plasmodium falciparum infection or Plasmodium vivax infection. The drugs which are in use against malaria, like if we go back into the history, it was the cinchona tree bark, which is also known as quinine, which was initially given for the treatment of malaria, which was during the World War I was replaced by a synthetic derivative of quinine, which is chloroquine. And chloroquine was effective for a long duration. However, the parasite in early 1970s, the parasite gained resistance to this particular drug, and this led to the introduction of the antifolets like sulfadoxine and pyrimethamine for the treatment of malaria. With time, the parasite has gained resistance to most of the drugs which has been used against the parasite, and at present, the artemisinin-based combination therapy, or which is also popularly known as ACT, is the best available treatment against the Plasmodium falciparum. And in fact, in northeastern region, there has been like artemisinin plus some other drug, like it's a combination therapy. So normally the artemisinin, as per our national policy, artemisinin along with sulfadoxine pyrimethamine is being given for the treatment of malaria. But due to the development of resistance in against SP in the northeastern regions, nowadays it has been shifted to artemisinin plus lumefantrine. And it has been recommended that the mixed infections should be treated as P. falciparum infection only. Primaquine is being given in case of Plasmodium vivax infection because this is the drug which can treat the hypnozoites. So basically, it is the primaquine is mainly given for the patients having Plasmodium vivax malaria. So, but as I said, that the Plasmodium parasite, right now, the artesunate based combination therapy is the main first line drug against malaria. But the problem which has come into picture is that now we are getting reports of the parasite also resistant to this particular combination therapy, where the reports have been coming from different regions of the world. So basically, all these drugs have substantiated a need to look into further novel drug therapies, or we have to look into the vaccine development of the vaccines, new vaccines against this particular disease. So, actually, what is drug resistance? When we talk about drug resistance, as per WHO, the definition of drug resistance is that the ability of the parasite strain to survive or to multiply despite the administration and absorption of the drugs in doses equal to or higher than those usually recommended, but within the limits of the tolerance of the subject, provided that the drug should gain access to the parasite or the infected RBC and should be there for a duration of time necessary for its normal action. So, when we talk about drug resistance, what is actually causing the drug resistance? Why the parasite is gaining the resistance against most of the antimalarial drugs which are in use? So, and from where this particular resistance has been coming up. So, if we look at the pattern of the resistance, or look at from where the first reports of the resistance have come in, we'll realize that most of the reports of this, or the chloroquine, if you talk about the chloroquine resistance, or we talk about the artemisinin resistance, most of this thing has come from Thailand. This is basically Cambodia Thailand border is the one, it is also known as the hot spot for the development of resistance, because most of the first reports of the resistance have come from this particular region. Okay. So basically, when we talk about drug resistance, it is mainly comes have more often evolved out of the areas of low malaria transmission. So basically, Thailand is an area of low malaria transmission, and once it comes into the picture, it spreads across to these endemic areas like Africa, where actually it is the worst thing, and where it is causing most of the mortality in the humans. But for us it might we might call it drug resistance, but for the parasite, it is actually the survival of the fittest, where it is survive, it is making the changes to survive in the given environment. So, what are the factors which are uh, responsible for carrying out the drug resistance? So, one, as I said, is the treatment failure, where most of the doses have been not properly regulated or due to the poor drug quality, which give rise to the drug resistance, or it might be due to the host immunity or cross multi resistance. It might be due to the genetic mutations which might develop in the parasite due to the drug pressure, which increases the gametocyte release or resistant mutant spread, or it might be due to the pharmacokinetics of the drug, where the drug is going, but there is an increased drug efflux, which is causing the parasite to gain resistance against that particular drug. If we look into the literature, we will find that the parasite has taken a lot of years to gain resistance against chloroquine. However, for SP and artemisinin, the resistance has developed in a very short span of time, because it is assumed that for the chloroquine resistance, there are multiple factors which are involved for the resistance to happen. And even if we talk in terms of artemisinin, the mode of action of artemisinin is actually it is working on different pathways, it has been suggested, and there is no single mode of action of artemisinin. So basically, all these things, the drugs have been effective for a small duration time, but later there was a development of resistance in the environment. So, what are the ones which are responsible for the drug resistance? So, while going across this, the people have found that there are certain point mutations in the particular genes which are responsible for the resistance. Like for chloroquine, it is the PFCRT gene or the PFMDR1 gene, multi drug resistance gene, transporter gene, which are responsible for the resistance. Like in the major mutation which has been implicated in chloroquine resistance is the K76T mutation, or a change of lysine to threonine at codon number 76 in the Plasmodium falciparum chloroquine resistance transporter gene. Similarly, there are different genes which are reported to be involved in the resistance against particular drugs. Like for quinine, it is PFCRT1, PFMDR1, and P-falciparum sodium proton exchanger 1. For sulfadoxine pyrimethamine, it is a point mutations in the DHFR and DHPS genes, dihydrofolate reductase and dihydroteropsynthetase gene. For artemisinin, it is being said to be KELCH13 or K13 gene. So there are different mutations which have happened in these particular genes, which can be utilized as a molecular marker for predicting the current status of uh, the resistance in the parasite population. So let's talk about these kinds of resistance one by one, and what we have found out from our study region, that is Bikaner. So when we talk about chloroquine resistance, as I said, that the mutations in the PFCRT gene are responsible for this particular resistance against this drug, and this particular transporter is located on the digestive vacuole of the parasite. This particular gene, PFCRT gene, is a highly interruptive gene with 13 exons and 12 introns present in between, and which only code for a 424 amino acids. Chloroquine resistance, as I amongst in this particular gene, there have been many mutations which have been reported, but K760 mutation is the one which has been clearly linked to resistance, chloroquine resistance. But however, it should be kept in mind that this particular mutation, K760 mutation alone, cannot cause the resistance, chloroquine resistance. This particular mutation, along with other mutations like a change of alanine to serine at 220 position or codon, are responsible for the resistance development. So, what this particular transporter is doing? So what happens is, if you are not having this particular mutations, if there is a normal lysine which is present in this thing, so as I said that this is present on the digestive vacuole, so this red thing is the red blood cell, and this is the parasite, and you can see this big circle is your uh, digestive vacuole. So this particular transporter is present here. So when we give chloroquine in normal condition, what happens is the base chloroquine is being taken inside, and what happens is, RBCs consist of hemoglobin. So what happens is that the parasite which lives inside the RBC digests this hemoglobin and converts it into uh, a protonated form and ferroprotofirin 9. Now this is basically uh, converted into a non-toxic substance. But what happens is, when chloroquine is being uh, so this chloroquine, what happens is, so this is in normal cases where it is a chloroquine sensitive parasite. So chloroquine blocks this particular polymerization into a non-toxic form, so that when this toxicity develops inside the digestive vacuole of the parasite dies off. However, what happens in a chloroquine resistant parasite when this is a mutant PFCRT, now this chloroquine, which is being responsible for preventing the transformation of the toxic form into the non toxic form, cannot do its function properly because this protonated chloroquine is being transported out of the digestive vacuole with the help of this mutant PFCRT. So now there is no accumulation of chloroquine inside the digestive vacuole, no accumulation of chloroquine. That means that this hemoglobin, the toxic substance which is being formed with the hemoglobin degradation, can now be converted into a non-toxic form, and thus there is no death of the parasite. So there are different mutations. K760 mutation is one of the mutations. As I said, it is completely linked with resistance. But other than this, there are different mutations like at the 74th position, 75th position, 220 position A to S change, R371I chain. So a lot of amino acids have been found to be associated with chloroquine resistance. Similarly, the other gene, the PFMTR1 gene or the multi-drug resistance 1 gene, which is present on the chromosome 5, there have been different mutations which have been linked with resistance, like N86Y, which is the major mutation, and 1034 codon, 1042 and 1246 codon, where a lot of mutations have been reported which have been linked with the resistance against the chloroquine drug. So when we looked at this particular mutation in the samples from Bikaner, and we found out that most of these samples were showing a mutant thing, like 96% of the isolate showed us the mutant phenotype, and only 4% of them showed us the wild phenotype, which means that the parasite population in the Bikaner region of Rajasthan is showing resistance against the drug in case of Plasmodium falciparum. And we also saw a predominance of SVMT hepatotype, where only a few isolates showed a normal wild type CVMNK haplotype. Okay. For the PFMTR1, we amplified a particular region from this gene, and then we went ahead for RFLP analysis. And if you see this RFLP analysis, a normal this thing where I'm seeing here 250 base pair band, uh, and all this, where I'm seeing this, in fact, I'm seeing this 505. This 505 base pair band only comes if it is a mutant phenotype. So you can see that we could also find the mutant phenotypes for the PFMDR1 gene. So overall, we have seen that there were many wild type, but a lot of almost, you can see that 19 samples were wild type for the codon 86. However, 11 samples were also mutant for the tyrosine residues. So, and we could also observe novel mutations like a serine to cysteine change at 134 position and serine to phenylalanine or tyrosine change at codon 232. However, as I have been talking about this, we have been talking about 5x. So when I looked at the orthologous gene of CRT, where I could see a lot of polymorphism or a lot of mutations in case of the falciparum, and when chloroquine was also given for the Plasmodium vivax cases, we wanted to see what is the impact of this drug pressure on the vivax parasite. But to our surprise, we saw that most of these uh, most of the samples, or in fact, all the samples for the PBCRTO gene, which was analyzed at that time, had a wild type genotype. Okay, so that means that chloroquine is still effective against this particular parasite. That means still, and in fact, in this particular region, chloroquine is still the drug of choice against the Plasmodium vivax malaria. Coming to the second thing, because the moment the chloroquine resistance came into picture, it was the whole thing shifted towards the second line of drug, which was sulfadoxine pyrimethamine, and which are the antifolates. So these particular antifolates have actually worked on the two enzymes of the folate pathway, the DHFR and the DHPS. It was realized that the mutations at a particular position in these two enzymes was responsible for the resistance against this particular drug in Plasmodium falciparum as well as in Plasmodium vivax. Like in Plasmodium vivax, the mutation at position 51, 58, and 117 were responsible for the development of resistance against SP. And it has been seen that at least three mutations needs to be there if the parasite has to be resistance against this particular drug combination. And the main mutation which has been responsible for this development of resistance is a change at 108 codon where a serine was replaced with a threonine, and this was actually responsible for alteration of the enzyme cavity, because DHFR is an enzyme and it has a very particular 3D structure. है ना? So what happens is uh, that the moment there is a problem or there is a mutation, the parasite changes its active site. And because of this, the drug is not able to bind. Am I audible and I'm clear? I just wanted to have an idea. And okay. So when we did a sequence analysis of this particular gene, we found that most of the isolates were double mutant. So you can see over here that this 100 position where we had a change from serine to asparagine, we could see this change in almost most of the samples which were sequenced, and this change was coming along with a change at 59 codon where there was a change from to arginine, while few of the samples showed the wild type genotype. In falciparum, the other enzyme which is responsible for the sulfadoxine resistance, which is also known as DHPS, we could see that the mutations which were there, we could only find four samples which showed the reported mutation at that time, where a glycine mutant glycine was seen at the in place of alanine at codon 437. But in addition, we observed three mutations, novel mutations at different positions, like a serine to phenylalanine change at 587 position, an asparagine to lysine change at 666 position, and a cysteine to tryptophan change at 668 position. So there were different genotypes which we could observe in a DHPS gene. And if we talk in terms of the predominance or prevalence of the DHFR and DHPS mutations, we'll see that most of the samples or most of the isolates were wild type for the DHPS gene. However, most of the samples were double mutant for the DHFR gene. So when we observe some novel mutations, we were eager to know whether these novel mutations have any effect on the drug binding or not, because we have come across that it's the major reason for the development of resistance in this particular gene is the alteration in the binding cavity. So we went ahead and we prepared a model, homology model for this particular um, enzyme. So DHPS, we prepared a model for wild type and we prepared a model for the mutant thing. And when we docked the sulfadoxine in the wild type PFDHPS, you can see over here that there were three hydrogen bonds which were formed in the cavity with the sulfadoxine molecule with wild type PFDHPS. However, when we did the same with the mutant PFDHPS, we could see only one hydrogen bond, which suggests that these mutations might be responsible for weakening the bonding between the drug and the enzyme. Coming to this, what was the scenario in case of Plasmodium vivax? So we also amplified these things from Plasmodium vivax, where the major mutations are again a similar mutation as 108 mutation, where we could see a change of serine to asparagine at 117 position, a change from serine to arginine at 58th codon, and a change from asparagine to isoleucine at 51 codon. There are other mutations reported also, and when we did an analysis of our samples from this particular region, we could see that there was not much of the changes over there, but however, we could see certain mutations in the nucleotides, and if we convert them into the amino acids, we could see that a novel mutation was observed at the 93rd position, where a serine was changing to histidine. In addition, we had this single mutant as well as double mutant in case of PVDHFR. Now, I want to emphasize here that in case of PFDHFR, we could only see double mutants. However, in case of PV, we could see all the things where majority of the isolates were showing the wild type genotype, and in addition, we could see a single as well as a double mutant in this particular case. And as Vivex is known to have different repeat regions also, we could also see a polymorphism in the tandem repeat region of PVDHFR, where most of the sample, while they showed a wild type thing, some of the samples showed a change in the number of the repeats which are present in this region. Like GGDN is the repeat which is present in this region. So if we talk about the what is the prevalence of these DHFR mutations, as I said, most of them were wild. However, we could see a single and the double mutant as well. And in case of only two reported amino acid changes were found in this thing in DHPS are reported, which is A383G and A553G, and we could see other changes as well in case of Vivex. And in case of Vivex, if we see, we could also see novel mutations here. You can see over here that we could see a change from F to L, M to I, and E to D. So these are all four other novel mutations which we could observe from the parasite isolates from Bikaner region. And when we did this thing, we wanted to look at the tandem repeat region in this thing. Like both DHFR and DHPS have this tandem repeat region in case of uh, yeah, sorry, DHF and DHPS has this tandem repeat region in case of Plasmodium vivax. So here also we could see the polymorphism in this region, and in this also we could see that most of the isolates were showing a wild type genotype. So again, we wanted to look into the thing that whatever novel mutation which I have just said here, we have found, are they having any effect on the drug binding? So we did a modeling and we found out that when we are looking at the docking between the normal uh, DHPS, wild type DHPS, and the novel mutations, we could not see much of the difference between the drug binding. And this particular thing suggests that this is only adding up to the polymorphism, and most probably it has nothing to do with the drug binding efficacy of the parasite. So this is the overall comparison where we are looking at the DHFR and DHPS gene from both Plasmodium falciparum and Vivex. So if we see over here that for DHFR in Vivex only it is most of the mutants, but for the other three like PVDHFR, PVDHPS or PFDHPS, majority of the isolates showed the wild type genotype. If we talk in terms of India, so I've told what was the scenario in our region of study, and if we talk in terms of India, overall India, we can see that in case of DHFR, PVDHFR, only in case of Karnataka, you are seeing the mutants, mainly the mutants. All of them are mutants, and in Tamil Nadu. So towards the south region, there were some level of the mutations. But if we talk in terms of the central India and in the northwest India, so we'll see that most majority of the parasites were kind of wild type. Assam, obviously, the northeast region is the region where most of the mutations are happening. So we could see that there also most of the isolates were having quadruple mutations. If we talk in terms of PVDHPS, here also the scenario is almost similar where there are mostly the wild type genotypes which are available for most part of India, except where in Karnataka and Tamil Nadu we could see a lot of mutations, and even in West Bengal. So we started off with chloroquine, went to SP, and then the parasite gained resistance to this particular drug also. WHO in 2006 recommended the use of artemisinin combination therapy for the treatment of malaria, where actually artemisinin has the fast acting drug which actually blocks the protein synthesis in the parasite and clears the parasite from the blood. However, the drawback of this particular drug is the very short half life. So, to maintain the effectiveness of the drug or for uh, treating the patients, it was recommended to go along with a long lasting drug like SP or the sulfadoxine pyrimethamine. So, after a lot of studies, it has been found that a gene or a locus on the chromosome 13, which is also known as KELCH 13, the mutations in this gene could be linked up with the artemisinin resistance, and the 3-point mutations which have been linked with artemisinin resistance are C580Y, which is the major mutations. And other than this, there is a mutation for arginine to threonine change at 539 position and a glycine to adenine change at 533 position. Okay, and a lot of mutations have been reported from different regions, suggesting that the spread of artemisinin is separate from different regions. However, many of these things are only predicted to cause, many of these mutations are only predicted to cause the resistance. Only these mutations, C580Y, is the one which has been implicated to be involved in the resistance pattern. So you can see over here A578S, all these mutations or C580Y. So I said C580Y is the main. This was suggested, this has been found from different regions. However, it has been shown that this is not linked to resistance. And if we compare the Vivex sequence, like if we compare the falciparum sequence and the Vivex sequence of KELCH 13, we could see that only there are 8 amino acid changes which are present in the propeller domain, which is one responsible for the resistance. So only 8 changes, amino acid changes, are present between falciparum and Vivex. And this A7578S, which is a change, which was a mutation which was found in case of Plasmodium falciparum, is one of those changes. So if I say in falciparum, an alanine is present at this 578 position, but if we see the corresponding position in case of Vivex, it is actually serine only. And even with the reports, it has been shown that this particular mutation cannot be linked with artemisinin resistance. During our studies from this region, Bikaner, we identified one novel mutations from in this particular gene KELCH13 propeller region where a valine was changing to leucine at a 487 position. And So we went ahead, we modeled this PFK13, and this PFK13 is actually, this propeller domain has six particular domains, blades, which are different, which here are shown with different colors. And when we try to link up our, see what is the effect of this mutation. So you can see over here, this is the wild type and this is the mutant type. And when we try to dock the drug between the wild and the mutant, we could not see any change in the binding up of the drug with the uh, protein. So it might suggest that there is no effect of this drug, of this mutation on the binding up of the drug to artemisinin. And when we looked the similar thing in Plasmodium vivax, we could not see any non-synonymous mutation from this particular gene. However, we could see a silent mutation in case of Plasmodium vivax at the 418 position, which was also seen from a very almost, which was also seen from very few samples. The work which, like the anti-folate drug resistant marker genes in Plasmodium vivax, where we have looked at the severe manifestation. So if I talk about in terms of the severe manifestation, what is actually the difference between the non-severe complicated samples and the complicated samples in terms of the mutations which are present in the isolates? I have not come across any particular mutations which I can say that it is linked with the severe patients which are showing the severe manifestations, and which is not present in the uh, uncomplicated cases. So basically, all kinds of mutations, if I see that a similar mutation pattern has been shown by both Plasmodium, I will say both the parasites which cause an uncomplicated malaria or the complicated malaria. Okay, and even in falciparum also the scenario remains the same. But if we talk in terms of the vivax malaria, Plasmodium vivax, one thing is clear from the studies which has been done across India and the world, that still the vivax, the parasite, is still not gained resistance to that level where we have reached in terms of Plasmodium falciparum. And so it's a high time that we should start using a proper dosage and start looking at novel or proper usage of the existing chemotherapeutics so as to prevent the parasite from gaining resistance, or where our efforts to treat the disease become ineffective. So basically, the thing which I want to emphasize over here is that we need to know more about malaria, and at the same time we need to take the doses or the medicines in a proper manner so as to combat the disease. Because with the national vector-borne disease program assuming to eradicate malaria by 2030 from India, it is our responsibility to work towards its eradication. Because if we talk in terms of the Southeast Asia, Sri Lanka has already been declared malaria free. So it's time that we need to look into, or we need to work towards making our country also malaria free. So this is our group at BITS Pilani. Our collaborators are from SP Medical College, Bikaner, Professor Dhanpat Koch and Professor Sanjay Kocher. And a lot of people have worked on different aspects of malaria, because drug resistance is one of those aspects. And these are our older PhD students, and this is our team at present who are working on different aspects of malaria. So the fight continues, and we are still fighting the disease. Thank you.
Thank you, madam. That was an amazing presentation. You work on PFDH gene and your compassionate traditional therapies, which gave us many insights on the topic. I hope that my research scholars, students, and also will be enlightened us. On behalf of the department, I thank you, and also behalf of the organizing committee. Also, thank you, ma'am. Thank you for your time. Thank you, ma'am.
Thank you.
Thank you, ma'am. Thank you for scheduling us on this date in spite of all things. So personally, I have to thank you and I have lots of gratitude for you. Really thank for spending time and giving a detailed lecture for this antimicrobial research, ma'am. Thank you. Thanks a lot, ma'am. Students, people can leave. If there is any doubts, people can post it in the query session, or you can unmute yourself and ask if at all any doubts are there. The queries that we have received doesn't have any major things to be questioned. So if there are no queries, we can close up the session. Shilvi ma'am, I think there is not much questions here. We'll end up the
Thank
session,
you.
ma'am.
Thank you.
Thank you, ma'am. Thanks a lot, ma'am.
Thanks a lot. Bye.
Bye, ma'am.